Good day, students. Today's topic of discussion is structure of cytoskeleton and functions of cytoskeleton. So, first of all, what are cytoskeletons? Cytoskeletons are basically the network of proteins which provide structural strength to the cell, support to the cell, and structural integrity to the cells. There are three types of cytoskeleton components present in the cells. The first type is microtubule, the another is microfilament, and last one is intermediate filament. So there are three types of components of the cytoskeletons are present in the cells. Now we will discuss microtubules very first. Microtubules were observed first time in 1953 by De Robertis and Frenchy in the year 1953, while the name microtubule was coined by Slaughterbeck. Now we will discuss the distribution. So cytoskeletons, they were found in five cells in the axis. The cells are nerve cells, mitotic apparatus, flagella and cilia, meristematic tissues of the plants, and elongation zone of the plant tissues, where the cells elongate after the cell division. Now we will discuss the structure of microtubules. So microtubules are basically they are the hollow cylinder structures. They are straight and their outer diameter is 15 to 24 nanometer. While their lumen diameter is 12 nanometer, it is basically a hollow cylinder structure, and their subunit, actually the subunit of this microtubule is protofilaments. These protofilaments further they are formed by the heterodimers of alpha and beta tubulins. Okay, so alpha and beta tubulins, they are the proteins together with some microtubule associated proteins, they forms the protofilaments and the microtubules. So now what is heterodimer? Actually heterodimer is the structure that is formed by the alpha and beta tubulin. They both together join and form the dimer. Here we will see the GTP binding site of alpha monomer is blocked at the interface of alpha and beta dimer, while the GTP binding site of this beta dimer is found at the open end of beta dimer. So in a head to tail orientation, there are many heterodimers of beta and alpha tubulin. They forms the protofilaments, and this filament, the protofilament, has the polarity, means one side beta dimer is always present, while on the another side alpha monomer is present. The site on beta dimer, beta monomer is known as plus or the fast growing end, while on the other end, opposite end, where the alpha monomer is present, it is known as minus end or the slow growing end. So here we can see there is the polarity present. In one side it is plus or fast growing end, and on the another side it is minus or the slow growing end. So 13 of such protofilaments, they join in a specific orientation and they forms a flat sheet, and that flat sheet further roll and forms this hollow cylinder structure which has a lumen inside it. Now we will discuss the functions of the microtubules. The first function of microtubule, it provides strength, shape and structure to the cells. The another function of this microtubule is formation of the spindle fibers during cell division. The next function is these microtubules, they are helpful in the transportation of ions and other substances in the cell, means intracellular transportation of the ions and other substances. The another important function of microtubule is the movement of cell organelles. Actually the cell organelles present in the cell, they cannot freely move from one place to another place, but there are some channels present in the cells, and on the determined specific paths these cellular organelles can move. These paths are formed by the microtubules. So here we can see that microtubules, they have the interactions with motor proteins, and these motor proteins, they interact with the receptors present on the vesicles or the cell organelles, and this complex now it can travel from one place of the cell to the another place of the cell. So this is very important function. The next function of microtubule is the formation of flagella, cilia and centrioles. Flagella, cilia and centrioles, they are formed by the microtubules. The another function of microtubule is the movement of flagella and cilia. Flagella and cilial movement is mostly happened by the microtubules. They are basically the cilia and flagella, they are made up of the microtubules, and therefore their movement also controlled by these microtubules. The another function of microtubule is cell division and elongation of the cells. Now we will switch over to the next type of next category, that is microfilament, and these microfilaments, they are having the width of 7 nanometer, and they are the twisted DNA like structure, and their fibers, ultimately this is made up of one protein, globular protein, and the name of that protein is G-actin protein. So this G-actin protein, it's a globular protein basically, but X-ray crystallography or some other techniques, it has shown that these G-actin protein are having two lobes and one cleft between these two lobes, and this cleft has the position to attach the magnesium ion and the attachment for the ATP. So many G protein molecules when bind to each other in a specific head to tail in a polar structure, they forms F-actin filament. So this F-actin filament, now we can see in the cleft side or the site where ATP bind, they forms the minus end or the slow growing end, while on the opposite side it has the plus end or the fast growing end. So depolymerization and polymerization process basically happens on the plus side of this F-actin filament. Two F-actin filament in a helical structure like DNA, they forms the actin filaments or the microfilaments. Now what are the functions of microfilaments? So microfilaments are important in giving shape, strength and structure to the cells. The next function of microfilament, they are helpful in the cytosolic stream. So in the cytoplasm itself, in the cytosol itself, the stream created by these microfilaments, and this is helpful in movement of the cellular substances. Microfilament have another function in cytokinesis of the animal cell. One more function of these microfilament is the movement of amoeba. So pseudopodia are formed in amoeba due to the microfilaments, and in one side the microfilament contracts, another side they relax, and this way they give motility to the amoeba, or they help in engulfing the food particles or any liquid substance from outside to the inside of the amoeba. The another function of microfilament is they are having the role in muscle contractions. So muscle contraction and for relaxation of the muscles, the microfilaments are very important. Now the third type of filaments, they are known as intermediate filaments. The intermediate filaments are they have some different differences from the this microtubule and microfilaments in such a way that the rigid structures, while microtubule and microfilaments, they are the flexible structures. So this is one important function. The another difference is that microtubule and microfilaments, they are having the sites for the attachment of nucleotides. In microfilament, GTP is attached. In microfilaments, ATPs are attached, while in intermediate filaments no such positions are present. The third difference is that these intermediate filaments, they do not have the structural polarity. Why it is so? If we see the structure of microfilaments, we see here that these microfilaments are basically formed from the keratin, mostly from the keratin proteins. And we see, if we see the keratin proteins, they have the N-terminal end, and another side it has the C-terminal, one polypeptide sequence. So on the one on this polypeptide sequence, in the one side N-terminal end, another side is C-terminal end. These N and C-terminal end, they are variable in their amino acids, while the in between part of this polypeptide chain, they have the conserved 7 amino acid sequence, and it is very conserved. So two such molecules, they are having two such polymers, polypeptide chains, they bind, intertwine to each other, and they form the dimeric structure, and they both have same orientation of N terminal in one side and C terminal on the other side. But they ultimately forms a tetrameric structure. So the another dimeric structure, it comes in the opposite orientation, and they intertwine. So here we can see that for one dimer it is N terminal and another dimer has C terminal. So there is no structural polarity here, because N terminal is present here, C terminal is present here. Here you can see C and N terminal. So this way they do not have the polarity. Such tetrameric structures, one after another, they joins and forms a long structure, and these long structures further intertwine and forms a rope-like rigid structure, and this rigid structure gives the strength to the cells. Now another important function of these intermediate filament is they helpful in attaching, encouraging of the cells to one another in such a way that if we want to pull apart one cell from the another, it is very difficult, because they are having the stitching of these intermediate filaments in between, and that's why the cell cannot be pull apart from each other. Thank you for hearing this lecture. I hope you will find it useful for you. Thank you. We'll meet in the next video tutorial.